Okay, so welcome everyone. Today we are here for a new series of the Actinobase talk. We have today Alejandra Prieto-Davó. So I'm going to introduce her. She studied oceanography in the Autonomous University of Baja California, where she also did her master in coastal oceanography. She did her PhD in oceanography also at Bill Fenical's lab. I'm sure you all met him, where she began also studying natural products from marine bacteria. She did her postdoc in microbial ecology, and she actually is working at the School of Chemistry at Universidad Nacional Autónoma de México, where she's a full professor since 2 years ago, leading the Marine Ecology Natural Product Discovery Lab. So we are very happy that we have her today to give her talk about microbial communities from the Maya underworld, sediment bacteria in Yucatan kinholes as a source of natural products. So if you have any questions, just post them on the chat, and at the end of the talk, I will read the questions to Alejandra, and we hope we have a nice discussion. So Ale, whenever you are ready,
Thank you very much. Thank you very much, Agustina. Thank you for inviting me to give this talk. I am very honored to be here with you today to tell you about the project that we've been working in our lab for the past 3 years. It's a project that started out of, as Agustina said, I work with microbes in marine sediments, basically, but I came to the Yucatan and then I was surprised by all of the natural beauties in this region. So I'm going to tell you about one of the most spectacular ones that we decided to work on. And I'm fortunate enough to hang around cool divers that can help us around to get samples from this place. So first of all, let me give you a quick view of where we're at, because we're very much away from everything. We're in this little town, fisherman's town called Cizal, which is in the state of Yucatan in Mexico, in the Yucatan Peninsula, about 60 kilometers away from the city of Mérida. That's the nearest big city. The town is only, like, as I said, it's just a fisherman's town. It has a very old pier, and this is the marine station down here to the left. There's the marine station that UNAM has, like to study basically all of the coast of Yucatan. This is what this station was created for, about over 15 years ago now, 20 years ago, I think, for the station, 15 years for the School of Chemistry being here. I've been here 10 years working already, and this is pretty much what it looks like. There's labs, there's coastal engineering labs over here, and so there's a lot of people from the School of Chemistry, the ecology, just the School of Sciences, and the engineering department of UNAM, and we all work together to try to bring more insight into what happens in all of these coasts, which was very much not studied when they started out this project. So Cizal might not be, and Yucatan might not be very famous for the UNAM's coastal station, marine station, but what we really are famous for is the Mayas, right? Everybody knows about the Maya culture, or most people know about it. They know about the pyramids, they know about their deities, right? Their relationship with the mystical, and we are also very famous because of the meteorite impacts that caused the dinosaur extinction. So here we are, right, in the middle of the Yucatan, and then boom, something hits us, and then it creates one of the massive extinctions on Earth. So that's what we're famous for. But what a lot of people don't really know is that the Yucatan is has the biggest or the largest underground aquifer in the world. Right? There are no rivers in the Yucatan. Everything is underground. And all of this water, and as you can see here in the map to the right, all of the water basically charges on the south central part of the peninsula. Everything percolates because this peninsula is made, it's basically a limestone platform that emerged about 30 million years ago, and it's made of karstic sediments. So that means that everything basically that rains and pours down into it. There's almost no filters. There's almost no soil in it. The soil is really, really thin, like the soil layer is very thin. So everything comes into the aquifer and it goes underground. So this creates major cave systems that are completely flooded and create this precisely these environments that I'm going to talk to you about today. And one of the interesting things is that there's basically flows that go everywhere because of the nature of these karstic sediments. And here's a couple of pictures. So this is the peninsula. This is the crater to the left. This is the crater where the meteorite impacted. So here we have marks of the crater, and all of these red dots that you see here are what we call sinkholes or cenotes. And I just read yesterday that there's over 8,000 cenotes around the whole peninsula, right? But there's a very big mark on where the meteorite created that crater, and there's a lot of cenotes around that area. So that's why it's called the Ring of Cenotes. But the interesting thing is that those flows go everywhere, and underground, the underground flows can change once they've eroded a part of the peninsula, and then they can move the other way. So this creates very local conditions that are very specific to each cenote that you study. And this is important. I'll tell you exactly why. Because it creates, first of all, this majestic and incredible places where we go and we can dive, right? And we go into the caves. These caves can be as huge as the ones that you see here. By the way, all of the pictures that I'm presenting here are Kai Vilchi's pictures. I'll show her Instagram in a second so that you can follow her. She takes incredible pictures. She goes with us in expeditions. And as you can see below all of this water, of course, just like in the oceans, there is dirt, there is sediments, right? So we decided to go with this group of people that were already exploring certain areas of the cenotes. They were looking at the flows, they were looking at the local fauna and at the very endemic species of different crustaceans. And so we decided to join them. This is a group called Cenoteando that I participate in with a bunch of people from CISAL that we go and explore everything we can about the cenotes. But my contribution was that we decided as a group in the lab to go and explore and see what was in those sediments and see what kind of bacteria lives in these sediments, what they're doing, right? And or what they could do. And then the Campeche, the state near Yucatan, that I see.
Alejandra?
Yes.
We seem to have lost you for the last like 10 or 15 seconds. Maybe you could just recap for a moment.
Okay, can you hear me now?
Yes.
Perfect. Okay, so I was telling you that thank you. That these places are amazing. So we decided to go explore and to see what was basically in the sediments of these caves, right? Because everything, as I mentioned, everything flows into the cave and then everything settles in the bottom. So we decided to try to see what kinds of microorganisms were here. This is another cave called Nomozon that we also explored. And so I was just saying that if you had a chance to come to Yucatan and explore the caves, you should do it. This is another type of cave. This is on the nearby state of Campeche. And what is super interesting about this cave is that in order to get to any water, you have to first go down in the cave 110 meters or so. So first you have to rappel down, because these stairs are definitely not safe. So everybody just rappel down. And these are the kinds of expeditions that are organized by the group in Cenoteando that I was telling you about before. And so they went down into the caves. This was a couple of, I think it was a month ago or so. And This is Efrain Chavez, who works with us. Sometimes he's now in UABC working as a technician, but they went into this huge cave that wasn't flooded, but then they found water. So he went and got us some samples from the sediment under there. So we went ahead and we submitted a proposal, and they approved to go around the Yucatan and try to get as many samples from different cenotes as we could to try to explore what was in them and see their potential for secondary, well, basically for their biotechnological potential, right? So we started doing the sampling. Unfortunately, of course, the pandemic hit us, so we haven't been able to go to more cenotes, but we have about 15 cenotes sampled, or 20, that we have taken a look at. And today I'm going to talk to you about 2 of those. There's a coastal cenote called Zapote. I'm going to go back a little to show you where it is. It's this one over here. It's close. Actually, it's closer. Zapote is closer to the coast. I don't know why the pin is all the way over there, but it's really close to the coast. And Nomozon is an inland cenote. It's on the south of Mérida. This white mark over here is the city of Mérida. And so Nomozon is in the southern part of Mérida, where that is also a very an agricultural and area where they also have a lot of pig farms, a very big farming area. So anyways, so this is what if you want to cross, like a cross section of El Zapote, which is the coastal one, would look like. It's about 54 meters depth, or that's as far as the divers could go. So we got samples from the area where there's a connection with the atmosphere, which we call the water mirror, and this is like the first area we sampled. Even though it's in the bottom, it still has sunlight hitting it. Then we call this area the transition towards the deeper, further end of the caves, called the cavern. And then we got samples from the very end over here called the caves, right? So we went and we sampled this area, and this is called hell bells, because these are some microbial like structures. They're made by microbes, and they hang from the top of the cenote. It's a really cool, very interesting cenote. And then there's Nomozon, which is the inland cenote, and we did the same. We did the water mirror sampling, the cavern, and the caves, right? This one's shallower, 34 meters, but it's also longer. So the divers went and get some samples, and we compared, and we definitely saw that the coastal sediment, and then this is all based on 16S, by the way. The first thing we did is we realized that definitely the microbial community, which would be this side, the left side of the graph here, had different abundances of all the groups, or of all the phyla, than did the non-coastal, the inland cenote, right? So as you can see, I mean, when you do the statistics, it shows that the 2 cenotes separate perfectly well. The coastal cenote is the red, all of the red marks, and the non-coastal is the black ones, right? This crosses over here is, we sampled outside both cenotes on the low forest of both areas, and the microbial communities are very similar. But we just wanted to know if there was any relationship between these microbial communities and the ones over here, right? So we saw basically that there's 2 different communities, one for each cenote, and that makes sense because each cenote has its own very particular characteristics, right? We also saw that mostly in the coastal, also you can also see it in the inland, but in this graph you can see it very well in the coastal, that it separates by the water mirror community, and then the cave and cavern look more alike. So all of that we've been studying, we realized that if you use the 16S and then go in the literature and see what kinds of microbes are present and what they could be doing just based on 16S, as I said right now, because we haven't finished with the metagenomic studies, but we're doing those too. But we saw that it changes from a basically sulfate reducing or sulfate involved community or sulfur involved community of microorganisms to a nitrogen involved community of microorganisms from the coastal to the inland. So those were very interesting results for us because we realized that the communities were very different, right? And that it's specific, and that's what we've been seeing, is that all of these communities are changing according to each of the cenotes we study. So which makes sense because, as I mentioned, physical characteristics of the places change as the flows start moving and eroding one part of the calcium carbonate, and then so the flow goes one way and then it can erode and go the other way, and it can bring all of these different I don't know how to say it, like chemical and physical characteristics to a different location and then affect the community. So that's what we're kind of seeing right now, and that's gotten us very excited. But we also took a look at what actinomycetes would be there, right? And to try to see what kinds of organisms we could recover from those sediments. And what surprised us was that there's a very high number of non-assigned or unassigned ASVs, right, in the non-coastal area. And as I mentioned before, this is an area where there's a lot of nitrogen in the environment, probably because of the farms, of the pig farms that are in the area, and all of the residues, right, from the farming that go directly into the aquifer, unfortunately. But that's how it works because there's no filter in the land, right? There's no actual dirt that it can go through and filter with the rest of bacteria. But then, so we saw that there was a potential here to isolate novel things and to take a look at them. And we also did the same with another cenote called Kayaking, which is also inland. And we have isolated a lot of microorganisms from this cenote with by many of the students that have worked in our lab. And once again, all of the work that I'm presenting here is done by all of the students and people that go in my lab. I just get excited about it and I put it up in presentations, but all of the work is done by them. And so what they started isolating all these kinds of microbes. We don't have a 16S ID on them yet, but we know that from the metagenome that there's definitely a big community of actinomycetales. So we're hoping that they're going to be, and the reason I'm presenting the Kayaking data is because this is based on metagenomics. The other one was 16S, but this is based on metagenomes, and they show that they have they do have a potential for secondary metabolite production. They do have a potential for biologically active compounds and some NRPS that I'm going to show you a little bit. These are all Corona, I think they're called Corona graphs done on MGRAS really quickly, just to show you that there are some, there is the potential for them to be producing some NRPSs and polyketides. So we got very excited with this data. And so we did what we like doing, which is going to the samples and getting isolates and try to see what grows. So this is basically stamping the sediments on agar plates and taking a look at what grows. And this is the kind of communities that are growing. There's a lot of fungi, but like regular fungi that are ugly looking. I'm sorry for the fungi people, but usually marine and water fungi don't look very exciting. But then we found these little guys, and there's all kinds of colors of these. There's these fungi that grow like little trees. There's green, there's blue, there's yellow. This picture shows very well how they come out of the agar plate, although it's not a very clear picture, but it shows. And so we took a look at those, and with the help of Dr. Wendy Escobedo, who's now part of UNAM CISAL also. She just started in January. She's a chemist, and with other chemists that I work with, Dr. Susana La Torre, of course, Bill Fenical, who's still, I'm still in touch with, and he agreed to also like take a look at these organisms, and Mario Figueroa at UNAM, who has been taking a look at the interactions between the microbes that we're finding in these sediments. And this is what I'm going to show you today. This is Alejandro Yam. He's Dr. Alejandro Yam. He's a postdoc. He was a postdoc in my lab, and he started looking at all of the compounds that are made by these organisms and he found a few of bioactive compounds in these microbes, right? Here's a picture of some of the microbes that we find in this collection. These are from different cenotes. These are the kind of things that we're looking at. And Mario and his students in Mexico City, because he works in the main campus in Mexico City, they did a very fast assay to try to see the interactions between the bacteria that we got from cenotes and some pathogens, and to see if there was any inhibition or no inhibition. So that was one of the essays he did. And then we ran an essay also, and he did so also in his lab. We did it in liquid. We grew the actinomycetes, the fungi, and then we grew them together. And we saw the compounds that they made during their interactions, and we saw how they can change. Like, for example, here, the ones on the left and right are the ones where are the organisms growing on their own, the microorganisms on their own, and then the middle one is the interaction, right? So we see things appearing, we see things disappearing, and so Mario has been taking a look at that. He's been looking at whenever there's inhibition or where there's pigmentation, right, or where something different happens in the plate, or when we see something cool in the flasks also. So he's been looking at UPLC, I think is what he's been doing with these microorganisms. And this is an actinomycete growing with a fungus, right? So this is actinofungus co-cultures, and you can see that there's each of them on their own, and things are showing up here, 11 and 18 on the fungus. These compounds are being made. I'm sorry, I'm not going to say them correctly, but I'm going to try. N-benzoyl lexophenylalanol and asparfurmate. And then the actinomycete made actinomycin D. So that was an actino that was active and against the fungus. And then he saw the interaction, and he saw certain things that are being made in more quantity. He hasn't identified these two peaks or the extra peaks that show up in the UPLC, but we're getting there. And also fungal or fungus co-cultures, same thing. He finds different molecules, and so we're looking at the interactions between things that are growing together in the sediment. So that's a very, very cool part of this project, because we see what kind of interaction they have with pathogens, but we also see the interactions they're having between themselves in their communities, right? So this is a very quick table of how this inhibition went. The dark blue is the areas where these actinomycetes, which are on the left column, actually had considerable inhibition, and the light blue is moderate inhibition. So we can see that they do have some inhibition against common pathogens. So that's also very interesting, and we're taking a look at that. So we're also in a collaboration, and this is how actually it all started. All of the cenote thing is I invited Sean Brady to give a talk here about 8 years ago, I think it was a long time ago. And we started talking about looking at the bacteria and using his platform to take a look at what was happening in the sediment. So we went ahead and we did this with Sean and his postdoc, I'll tell you, Vincent Leaves, and he's working with Sean. And so what we did is we went and we got all the DNA out of these sediments, right? And then we followed his small molecule discovery pipeline, which is basically amplifying the KS domains and the A domains for PKS and NRPS. And we would analyze the results to see which samples had the best potential to go explore and do more work on them, right? Both cultivation dependent and cultivation independent. And this is what I'm showing you here. This is what Vincent Leavis has been working on in the past couple of years. And he has a new emulsification paper coming out soon, I think in the next couple of months, where we not only get the KS or the A domains, we get bigger fragments of the biosynthetic cluster. So he's been working on that with the sediments from all of these cenotes. And if you see on the left side, all of the colors on the left represent a different cenote. And we definitely see this is the diversity of KS domains plotted against each other to see how they group. And we can see outliers here, mainly Tsa, which is the red one, the blue one here called Zapote, which is the one I talked to you about from 16S and how interesting it was. And then this one right here called Polak. Also these three, because they're all kind of the same system, because they're interconnected, so they also group over here. So we went ahead and we also see, and the B panel is where in the cenote do we see these groupings. And basically it's the blue and the orange for the inland cenote, which is cavern and cave. And that area of the cenote groups over here on its own. And for Polak, it's the whole cenote, and same for Zapote. So that the ones that are influenced, they're the coastal cenotes, which are Polak and Zapote. It's just that Zapote is on the Caribbean side and Polak is on the west side. It's on the Yucatan area, along the state of Yucatan. Very important, and I forgot to mention, is the coastal cenotes are influenced by seawater inflow, because everything is underground, right? But there's also a connection to the ocean on the coastal area. So these are like underground estuaries, and so the water flows under, of course, the fresh water, but there's interaction with both flows. So that makes these locations really, really interesting. And when we see the diversity or the estimates, we see that these cenotes are interesting. The black bars are, interesting enough, all of the samples from outside, from the forest. But when we see what is inside in the cenotes, we see that Polak is one of them, Tsa and Zapote, right? So they're interesting locations to take a look at, further look into. And these are the kinds of molecules or that the KSs were related to. So we know that these microorganisms have a good potential of making natural products. When we take a look at what kinds of molecules and where they're present, we see that for Polak, this is all data for Polak. I don't know if I put it over here because this thing is, no, I didn't put it, but this is for Polak, for the cenote Polak. And so you can see that there's different things like curacin, myxothiazole, Jamaicamide, all of these KSs that are related to these known compounds are present in these sediments, right? But also, and this is a figure that Pablo Suarez-Mo, he's a postdoc in my lab right now, and he took a look and he did, using NAP2, he basically did the phylogenetic tree to see what kinds of things were present in our samples, and everything that you see here colored, he's colorblind. So I'm going to apologize for the colors in this graph, but he's colorblind. So he needs to make it like really contrasting for him to know that he's doing it right. So I appreciate that about him. And he, so he made this. And so there's a lot of KS is related to compounds that have anti-cancer activity and also antimicrobial activity. So that is very exciting to us, because we know that we're looking in a good place for all of these kinds of compounds, right? So we went back, everybody vaccinated finally, and we did a sampling trip out to Polak, which is about an hour away from Sisal on the coast. So you have to go down to the west side of Sisal, to the southwest. And once you get to the area, you have to hop out, and then you have to kayak for another hour to go into the cenote, because it's inside, right? It's in the coastal area, in the mangrove, but it's inside. You have to go. So we got everything packed, we got everything ready, and we started kayaking into this sinkhole. And this is what the kayaking looks like. You gotta go in like very narrow places full of like of these mangrove roots, and then you come into this huge area that is open, and it's a great place to go to get to know all of the wildlife. We saw snakes, we saw birds, we saw a lot of fish, a lot of different things, a lot of spiders. And so there, everybody got ready, and the divers went down, right? This is again Efrain Chavez and Luis Lievano, and this is Kai Vilchis. She's the photographer I was telling you about. So they went down and got the samples. They're certified cave divers. So in this one I stayed up. I didn't go down with them. I usually go down on the first one, but this is just like a barrel down to the bottom. So they went down and they got some of the sediments from the different depths, and I think it was up to 30-meter depth that they went down to. They brought back all of the samples, and we were very excited to get, of course, mud as always. So this is what our collection looks like when we come out of those places. Everybody is really, like, all of the people that bring us to this place get really excited, and they're like, "Oh, you wanted mud." And we're like, "Yeah, we wanted mud." So what we're doing right now, what's happening at the moment as we speak in the lab, I hope so, is we're doing cultivation independent work at this lab. This is headed basically by, as I mentioned, Pablo. He's the postdoc right now in my lab, and Jose Carlos, he's another postdoc, and Perla, she's a PhD student, and Perla and Jose Carlos are doing metagenomic libraries to try to look for the activities. We're doing both PCR or probe hybrid. It's basically, we're going to do PCR to see which clones have what we are looking for, and also activities. So also the function-based screening. So that's what we're doing right now. I don't have any data on this, but that's what's happening right now in the lab. We're getting these libraries to work, and then Pablo is doing all the metagenomic mining. He's looking at the metagenomes from these places, and he's trying to look for the biosynthetic clusters. These places have an amazing amount of microorganisms. They're really diverse. So we're also doing some experiments to try to reduce that diversity and try to get bigger contigs and better resolution of the metagenomes. So we also have, of course, thanks. Oh, I forgot to put the credits on these pictures. These were all by Wendy Escobedo in the lab. She took pictures of the microbes we're working with. She's, as I mentioned, she's the chemist now that's working with the extracts from these microbes, and we're very excited because we're getting, like, so far we've isolated about 40 colonies, not much, but enough to start working with. That there's 15 gram positive that have shown inhibition or pigment, and then 4 gram negative. There's not a lot of gram negatives, but there's 4, and they show inhibition against something in the plate or pathogens, or they have some kind of pigment. Those are the things that we look for. And this is what the colonies look like. I posted a couple of these yesterday in our Instagram account, and but they're looking good. We're getting there. We're exploring. We're exploring as much as we can from all of the aspects of microbiology that we can think of. And basically on the mining, right, there was this Maxi Medema published this recently, a couple of weeks ago, on how bacteria would be the best target to look for metabolites and genes, right? And fungi also. So bacteria and fungi would be a very good place to look into. So that's what we're looking at right now. We're working with fungi with Mario, and bacteria also. And then Wendy, Bill, and Susana, everybody's involved in looking into these bacteria and trying to look for these biosynthetic pathways. This is just an example I had here for just purpose of showing how all of the domains come in modules, and then they are coded for different genes, right? So I'm not going into detail with this with you guys, because I'm assuming that you already know this part, but this is just for educational purposes on how these biosynthetic gene clusters are established inside bacteria, and they're all together, which is great. So we can look for them by genome mining. So this is basically what we're doing right now in the lab. We look at marine and cenote sediments. We look for novel natural products. This is a small sketch that has to be finished because we're looking for the molecule here on the red dot. And what we do is we go out, we explore sediments, we explore sponges. We found cave sponges in these caves, and we're taking a look at them, and we're generating libraries. We're isolating all of these microbes, and we're trying to see if there's anything new in these places, just because of the ecological niche that we're exploring. Nobody has ever gotten samples from these places before. So we're hoping, and we're keeping our fingers crossed, that we will have a lot of new things to work with. And as I told you before, this is all done by a group. I just have the privilege of sitting down here and telling you about it, but this wouldn't be possible without all of the students that come to scientific stays. They're either summers or 3-month stays, or they come and work on their thesis. It's very hard for us because we're so far away from everything. It's hard to get students to come and stay, but we've been doing it. So we've been fortunate enough to have students that have worked. And right now we have PhD students. This is Norma Angelica, we call her Angie, and she is the tech in our lab, and she's the one who helps all of the students move around and tell them exactly where everything is. So she's basically the boss around the lab. And Adan is a PhD student. Ninette is an undergrad student. Perla, I just mentioned, she's doing also her PhD. Wendy is our new faculty in the School of Chemistry in CISAL, and the 2 postdocs. So, and this is the group of part of the group of the divers that I work with. They're, as I mentioned, we're part of a big group called Cenoteando. Over here is the website if you want to take a look at it. The Polac expedition video is in that website. If you have a chance, go ahead and look at it. And basically, that's what we're doing in the lab. That's what we've been working on for the past few years. And we're getting very exciting results. And hopefully, soon enough, I will be able to show you all of the cool molecules that we get from these novel environments and their microorganisms. So thank you very much for your attention. I hope you enjoyed it. And I am open for any questions.
Thank you so much, Ale. What a wonderful talk. I really love your photos. You have very cool actinos. So wonderful, just wonderful.
Thank you. I have to thank Wendy for those, because I was like, please send me pictures of actinomycetes or anything that grows on the plates.
Yes. So while we wait, because we have a small delay with the YouTube channel, and we hope we see a lot of questions there. I have a question, but it's just from my curiosity.
Okay.
Because you know that we work here with marine sediments, right? So I just wonder, how are they different, like the cenote sediments from the marine sediments, in terms of chemistry and physical and also bacterial communities? Because I know that you know a lot of oceanography. Okay. So yes, can you explain me?
Yeah, in these sediments, the biggest difference I've noticed is the amount of organic matter. Like up to 50% of the sample can be organic matter, which doesn't really happen much in the sediments in the coastal area here. The coastal area in the Yucatan is mostly calcium carbonate, and there's nothing in it. It's just really white powdered sand. It's very pretty and beautiful, but there's almost no organic matter in it. And these sediments are full of organic matter. Let me think what else I have seen on them. Some of them have a lot of, as I mentioned, they have a lot of nitrates compared to.
So you use like special media to isolate the bacteria that comes from the cenotes, like in terms of the marine, the media that we use for
the ones. Well, no, what we do is we have been reducing the amount of nutrients in the media, because we've noticed that they grow better when we put less in it. So we're using the same formulas, and we're actually the ones that came from Polac. Jose Carlos over here is the one who's been working on those, and he's been using different media. We're just trying we're very new at trying to figure out how to grow novel things from these sediments. So he just used a bunch of different media to see what he could get. So we don't just use the regular A1 that we call it, which is peptone, yeast extract, and what else?
Very rich, right? Yes.
Right. The one that we use for actinomycetes. But we have seen, so from other studies that hopefully will be published soon also, we've seen that we did a metagenomic study to see how this media affects the community. And we've seen that instead of making instead of increasing the diversity of actinomycetes, it actually reduces it. So we're trying to come up with better ideas on how to find more things in the actinomycetales. So yeah, but what else can I tell you about these sediments compared to the communities? Yeah, the communities change a lot locally. So each cenote looks like since there's very different physicochemical characteristics in these sediments per location, just because the flows go everywhere they want and they change all the time. So it looks like every community, and it's very different on its own.
Like even in the dependent, yes.
Even in the same cenote, the one that is influenced by light is very different from the one inside the caves. So, so yeah, we see that there's a big difference.
Cool. So I see that we have our first question. It's from Lorena Fernandez. So she says, thanks, Alejandra. Fantastic talk and really cool work. Have you got a favorite strain from your collection, and why is that? I'm sure you have a lot of
Well, no. Okay, so here's the thing. Not yet. Unfortunately, not yet. Because we weren't not just hit by the pandemic. Last year, we had 2 hurricanes and 2 tropical storms that completely flooded the access to our labs. So for about 6 months, we just couldn't go in. So we're behind a lot. And with all of these pandemic online classes and everything, I really haven't been able to go to take a look at everything in the lab carefully. So all of the students and the lab techs and Wendy have been able to go to the lab more often than me. So I haven't actually looked and said, oh, this is my favorite. But usually my favorites are the ones that have really bright colors. So I'm pretty sure I'll find one of those.
I'm pretty sure you will find a lot, and then you will have a lot of favorite ones.
Yeah, thank you.
It's hard when you have a lot of cool strains and then you have to select one. So then it's another problem, how you prioritize those cool strains, right?
Right, exactly. Well, we're doing for now on those, since the first 41 are the ones, and we have more than 41 from the cenotes. It's just Polac has this. We've been isolating this 41 from Polac, but we've had other strains from the other cenotes, and we have over 250 strains right now in our collection. So we do have a lot of them, and we're doing right now. The next thing to do is doing just 60 nests to see what's in there. But we've been very slow at getting there, unfortunately, because of all the restrictions on going into labs. But we're getting there. So hopefully this year, by the end of this year, we'll have all of that information. I see a question on what we use for actinomycetes. We've been basically using, we've been using the same medium that we use for marine actinomycetes. We just use regular tap water, or we go, since we have access to not the cenote, but the mangrove areas, right? The cenote takes a while to get there, but we have access to the mangrove area. So we go and get bottles of gallons and gallons of water from the mangrove area. And we use that water, we autoclave it, and we use that water to grow things too. So that's helped a lot also.
Cool. So I see that there is no other question. I think in the chat, let me check. Yeah, Lorena says, no worries, you will have to tell me about your favorite strain when you find one. So yes,
I will probably give you a whole talk on my favorite strain from the cenotes next time.
We all want to know more about it. So I have one more question, just because I'm very interested in your work. So you told us about some actino and fungal co-culture, right? And then you did also fungi and fungi co-culture. But are you planning also to do actino co-culture?
Yeah, we could do that. We have done interactions, not a co-culture in its own, but one of the first screenings that we do is we basically grow, we streak the bacteria that we have, and then we streak them one way, and then the other we just do do a grid of the bacteria that we have, and then we see if there's any interactions. And yes, so with that, we've seen that there's actinomycetes that promote sporulation on another one, or that inhibit the growth, or that make the other one produce a pigment just where the area where they touch. So it's very interesting interactions amongst them. We haven't done the co-culture to look into the extracts, but yeah, I'm sure that Mario and Wendy would have no problem into looking at those. Actually, Wendy is the one that's going to be doing all of those projects right now. She's going to be the chemist in charge of all of these strains. That's why she has so many cool pictures. She's really good at working with actinomycetes and bacteria, and so she took these last week, and they look really cool.
Yes.
So I'm pretty sure she's excited about working with all of these extracts. So and we're very excited to have her around too, so that we can see more about what's going on in between these microorganisms in the sediments.
So we look forward to your next talk, Ale. I think you just got us all motivated to listen to your work.
Thank you. And I hope you can come and visit when things get better everywhere around the world and we can all travel again. It would be great if you guys could come and see these places, because they're amazing. As I mentioned, just go and see Kai's account. She's an incredible photographer, and she has these cool, I'm gonna just make you dizzy for a second to put those pictures again. Mm-hmm. But she has these really cool pictures of places that are amazing. Like she goes, it's just going into them and then checking it out, and being in a cave but underwater is just, it's just amazing. There's also these ones that are super little and tight. Of course, I don't go into them, and they're crazy that they do. But we do have samples from very tight places that nobody has gone ever before. So we're very excited about looking at those bacteria and seeing what's in there.
Cool. So thank you, Ale. I think that we don't have any further questions, and I am not going to ask you anymore.
Well, thank you very much for inviting me, and thanks for letting me tell you about what we've been doing in our lab.
Thank you for the cool talk. So we hope we can see how this is going, like in a year or so.
And you have now my information. I put it in the talk, so you can take a look at it, and you can email me if you guys are interested in anything in particular. So please go ahead and contact me.
Cool. Thank you. Thank you so much. And thank you everyone for watching. So we meet in 2 weeks.